And thank you for joining me today for the Biodiverse Festival in 2021. I'm a PhD student at UC Santa Cruz and also worked on this project in collaboration with the Cheadle Center for Biodiversity and Ecological Restoration. Thanks for joining me for my talk titled Restoring Endangered Species, Lessons from Lupinus nipomensis, Reintroductions 5 years later. I want to take a moment to do some acknowledgements, especially for my collaborators, Lisa Stratton, Lindsay Roddick, Christy Scarazzo, Connie Rutherford, Kat Darst, and Wayne Chapman, as well as our funding sources, and all of our other partners and field assistants that made this project possible. I also want to mention that this work took place on the unceded territory of the ancestral Chumash people, which were forcibly removed from their lands. Today, the Chumash are working to heal and decolonize from historical trauma. Given that we are talking at the Biodiverse Festival, I thought it'd be appropriate to talk a little bit more about why biodiversity and endangered species are important. First off, we know that global extinctions could lead to up to 10% in reductions of species per year, and that's just a conservative estimate. And in likelihood, we may actually experience greater loss, and this loss may accelerate as climate change accelerates and there's more cascading effects. Endangered species are important because they can provide a lot of specialized interactions, some of which have never been studied and are less understood. And so removal of some of these endangered species could eventually lead to extinction cascades that could lead to extinction of species that rely on them or regularly interact with them. These losses of species eventually may lead to reductions in ecosystem services, such as pollination services, carbon storage, and even potential utilitarian uses for humans in the future. Endangered species are important for many different reasons, but why is reintroducing or restoring them so hard? Well, there are several reasons for that. One of them could be because we lack sufficient funding to conduct dedicated monitoring of some of these existing endangered species populations and their subsequent experimental populations. This creates a dearth and lack of knowledge in natural history, which sometimes is considered these days as a lost field, but it can be really, really important in endangered species reintroductions because it can tell us a little bit more about what periods of time and climate this endangered species persisted in, what other plants occurred nearby, and potentially other types of insect interactions that could have occurred with the plant. In other cases, it may be hard to restore species because we've lost a lot of their historical habitat. And the existing populations that we currently see could be the peripheral habitat of when the population was not endangered, and maybe just an area where the population can persist but not spread or grow. Finally, we often do not necessarily consider the spatial heterogeneity at small-scale local site restoration or reintroduction efforts. And this again is due to a lack of funding. However, microhabitat preferences and conditions could be really important in understanding why certain endangered species project reintroductions succeed and while others tend to not necessarily reach as high standards. Today, I'll be talking about Lupinus nipomensis as a case study for endangered species reintroduction efforts. Lupinus nipomensis is a Fabaceae plant found in the back dunes of San Luis Obispo County in California. There are often less than 1,000 individuals found per year, and it's annual, and so population can vary interannually, especially depending on the weather and climate. Because it's a Fabaceae plant, it's a nitrogen fixer, but also has a tendency for self-fertilization. And based on other field pollination studies we've done, we are leaning towards it being able to self-fertilize. And an important point in that is that plants that are able to self-fertilize, especially endangered species within small species ranges within fragmented habitats, often suffer from greater genetic inbreeding depression, which could eventually lead to population declines and potential local site extirpations. In order to better understand rare species reintroduction efforts, we used a series of 3 experiments at 2 different sites within the local area the species would have occurred in. We first established reintroduction plots in 2015 to assess the microhabitat, such as the abiotic factors that could affect temperature or soil moisture, and also the effects of caging on reintroduction efforts. We also set up additional plots in 2019 at the original site and at one new site to understand biotic interactions a little better and to test whether or not the effective caging was beneficial due to fogging and shading or potentially through herbivory reduction. And then we were also interested in the impacts of competition. To understand the responses of Lupinus nipomensis, we assessed its annual germination, annual reproductive output through counting seed pods, and assessed its relative herbivory severity. To start off with, I just want to mention that for this slide and the next slide that I'll be talking about, the data being reviewed is from the 5-year data set up from the original 2016 plots. We found that Lupinus nipomensis success was strongly influenced by microhabitat preferences, especially those that could affect abiotic conditions. For example, we found that the total germination was strongly affected by the topographic slope the plots were located on, and that steep and swale slopes often had higher germination, and especially swales. We found that the aspect, on the other hand, was really important for seed pod production, and that north-facing slopes often produce much more seeds than south-facing slopes. And interestingly, this trend was only for seeds and not for germination, whereas the topographic slope effect was only for germination and not for reproduction, indicating complexities in reintroduction efforts and selecting sites for either germination or reproduction. We found that caging strongly improved the success via germination and reproduction, as you can see on the graphs in the upper right, where we compared caged and uncaged plots for germinants and for seed pod production. We found on the bottom plot that seed pod production benefited from mild herbivory compared to severe herbivory, and severe herbivory often decreased seed pod production. Caging increased also the likelihood of mild herbivory and decreased the likelihood of severe herbivory. In this slide, I will be touching a little bit more on the 2-year data set that we recently set up in 2019, which tests for the effects of herbivory and competition, also known as biotic factors. So we found that caging strongly influenced the success of Lupinus nipomensis individuals. However, because this area has coastal fog, the cages could have provided fog drip to the plants or potentially provided reduced exposure through shading. And so we wanted to see whether or not the positive benefit of the cage was due to this structural shade fog drip type of influence, or if it was just due to reducing herbivory. We did this by setting up lifted cages, as you can see in the upper left-hand corner, and control cages, which were unlifted, similar to our original cage design. And we found that caging and lifting of the caging had significant effects, indicating that herbivory was a strong limiting factor. On the bottom right-hand corner, you can see the 2 plots related to the lifted cages. On the left-hand side, you can see that germination is greatly improved with cage plots. And on the right-hand side, you can see that seed production is severely reduced when the cages are lifted and potentially allowing in more herbivores. In the upper right-hand corner, you can see some plots where, from another set of experiments we did, testing for the influence of competition. In these plots, we weeded some of the plots and removed all existing species in the plots, or we kept it as a control and did not do any weeding. In this instance, we found that weeding had no strong effect on either the germination count or seed pod production for Lupinus nipomensis. So what does this all mean for Lupinus nipomensis and endangered species recovery at a greater scale? Well, from our results, we can tell that endangered species reintroduction efforts are often complex and may require multifaceted approaches. We found that Lupinus nipomensis had different selection factors for germination and for reproduction, indicating that future efforts will need to balance these types of successes and ensure that not only are we getting germination, but also getting seed pod production, which is necessary for the resilience and continuation of the populations. We also know that understanding natural history and microhabitats are really important for improving success. For example, we found that germination was preferred at swale sites and reproduction was highest in north-facing slopes. And these types of areas only occur in 10% of the original outplanting site that we chose. And so if practitioners were to just go out and restore and not account for spatial heterogeneity, they may see much lower success than we find in more concentrated, specific microclimate areas. And we may also find that abiotic and biotic factors may interact to further complicate recovery efforts. And so, for example, right now we have been able to tease apart that the caging has reduced herbivory and is not benefiting the plants because of fog, but there could be other complicating factors that we may need to think about in the future, or other efforts we'll think about. For example, how will climate change affect the ranges of competitive species or pests and diseases that may further influence the outcome of reintroduction efforts? So to finish off, I just want to end with some potential recommendations to help improve endangered species recoveries in other areas and for other species. First off, be sure whenever you're undertaking a project to really understand and research the natural history of the species you're working with. Natural history, again, is sometimes thought of as a lost field, but is really important to really understand the historical patterns of where the species occurred, when it occurred, and how did it interact with other species in its area. It's also important to assess site heterogeneity, because as we found, Lupinus nipomensis only could occur and really thrived in a very small subset of the types of habitats we introduced it into. And so if we spent a little bit of time and money assessing that, we could potentially increase the return in the future. It's also important to determine the different factors influencing germination and reproduction, because as we saw in our study, reproduction and germination were affected by different factors, but both are important for the persistence of the population. Finally, conducting smaller focus studies can help reveal mechanisms of success and of failure, and why some plants may be producing less seeds or germinating less, and can be really informative for future outplanting efforts. With that, I just want to thank you all again for coming and choosing to watch my talk at the Biodiversity Festival in 2021. My name is Justin Leung. I am a graduate student at UC Santa Cruz, and you can reach me with any questions or comments at my email, jluong4@ucsc.edu, or you can follow me on my Twitter at Justin C. Leung, and that's spelled L-U-O-N-G. Thank you again for coming, and have a great day.